Welcome to This Explainer. Today, we are unpacking a seriously fascinating chapter of biology. And the crazy part is, it's happening right in our own backyards. We're talking about the highly evolved, honestly kind of mind-blowing mechanics of sexual reproduction in flowering plants. What might seem like just a simple, beautiful part of nature is actually this intricate world of survival, high-stakes strategy, and microscopic engineering. So, let's jump right into it.
So, I have to ask, are flowers just here for our enjoyment? I mean, for thousands of years, we've used them for aesthetics, for perfume, as symbols of emotion, right? It's super easy to develop this human-centric view and just assume all these vibrant colors and amazing scents are entirely for us. But nature rarely expends that kind of energy just to look pretty.
Yeah, biologists, they have a very different perspective. As the text puts it, flowers do not exist only for us to be used for our own selfishness. To science, a flower is literally a morphological and embryological marvel. Every single petal, every hue, it's a calculated evolutionary adaptation. And it's all designed for one absolute bottom line purpose, to ensure the formation of seeds and fruits, which of course guarantees the survival of the entire species. To really wrap our heads around these mechanics, we have to look at the flower's blueprint. There is a super strict division of reproductive labor going on here. You've got the male structures, the stamens, with their pollen-producing anthers, and they sit right in proximity to the female pistil, which features the receptive stigma and the ovary down at the base. Think of these as highly specialized factories. They're forging gametes long before a bee or a butterfly ever even arrives.
Okay, so before a plant can reproduce, it obviously needs to prepare those gametes. And it does this through two parallel simultaneous journeys. On the male side, we have microsporogenesis. Deep inside the anther, sporogenous tissue undergoes meiotic divisions to form microspore tetrads. These eventually become your pollen grains. Meanwhile, on the female side, you've got megasporogenesis. Down inside the ovule, a single, frankly massive, megaspore mother cell also undergoes meiosis to produce 4 megaspores. But, and here's the catch, usually only one of these actually survives to become functional. The absolute key takeaway here is that both systems are locking into the strict, highly regulated cellular division just to prepare for the main event.
Now, if we follow that one surviving female megaspore, it reveals a genuinely fascinating biological anomaly. This functional megaspore starts dividing mitotically, but these divisions are strictly what we call free nuclear. What does that mean? Well, the nucleus divides into 2, then 4, and then 8, but cell walls don't immediately form around them. So for a bit, it's just one giant cell with multiple nuclei just floating inside. That is, until they finally organize into the mature embryo sac. And that organizing leads to a highly specific, totally fascinating structural outcome: a 7-celled, 8-nucleate embryo sac. Let's break that down. Inside, 3 cells form the egg apparatus, along with its supporting synergids. 3 more become antipodals at the complete opposite end, and then one massive central cell holds 2 polar nuclei. So count them up, 7 distinct cells, but 8 nuclei in total. It is a perfectly primed biological machine just waiting for the go signal. Now, before we get to the actual fertilization, I have to share a pretty surprising real-world fact from our source chapter about the male counterpart, the pollen grain. See, pollen isn't just, reproductive dust. It is practically a biological superfood. It's incredibly rich in nutrients. In fact, it's actually become really popular in Western countries to consume pollen tablets and syrups as food supplements. People claim it increases athletic performance and, get this, even the performance of racehorses. No way, right? But it's true. Nature packed these tiny grains with literally everything they need to survive their journey. And humans definitely took notice. But of course, the true destiny of pollen is not a dietary supplement for a horse. It's the stigma of a flower. This brings us to the real climax of our chapter, the pollen-pistil interaction. You see, when a pollen grain lands on a stigma, it doesn't just automatically fertilize the plant. Nope. It triggers this critical, high-stakes chemical dialogue. The pistil essentially interrogates the pollen grain. It's asking, are you the right species? Are you the right genetic match? If it's incompatible, the pistil completely rejects it. But if it gets the green light and is accepted, we move right into the action sequence. Once it's accepted, the pollen grain germinates and sprouts this tiny pollen tube, and this tube physically grows all the way down through the tissues of the style just to reach the ovary. I mean, think about the incredible navigation required there. The tube is actually guided right into the synergids by these special cellular thickenings called the filiform apparatus.
So the tube is acting as a delivery vehicle, carrying exactly 2 male gametes right to the doorstep of the embryo sac. And this sets the stage for steps 3 and 4, which, by the way, are completely unique to flowering plants.
We call this phenomenon double fertilization. Remember how that pollen tube delivered 2 male gametes? Well, they both have a job to do. The first male gamete fuses with the egg cell. We call this syngamy, and it creates the diploid zygote. That is what will eventually become the brand new plant. But what about that second gamete? It actually bypasses the egg entirely and fuses with those 2 polar nuclei sitting right in the middle of the central cell. We call that triple fusion, and it creates a triploid cell. So, because there are 2 distinct fusion events happening inside the exact same embryo sac, it's called double fertilization. Honestly, it's just an absolutely brilliant evolutionary strategy. Right after this double fertilization, the flower undergoes a massive transformation. The diploid zygote begins to divide and grow. It progresses from a simple proembryo into a globular shape, then to a heart-shaped stage, eventually maturing into a full embryo complete with cotyledons. The plant is essentially taking a fully formed baby plant and packaging it securely into a protective casing, the seed.
But here's the thing. A baby plant needs a packed lunch, right? It needs energy. That is exactly where that second fusion event, the triple fusion, comes into play. It develops into the endosperm, which acts as the built-in food source for the growing embryo. And there's actually a fascinating everyday example of this from the text that you have probably tasted. Endosperm development often starts out as free nuclear, which means thousands of nuclei dividing without any cell walls. Think of a tender coconut. The sweet liquid coconut water you drink, that is actually pure free nuclear endosperm. Later on, cell walls start to form around these nuclei, creating cellular endosperm. In a coconut, that's the solid white meat or kernel that you eat. So, literally, the next time you sip on some coconut water, just remember, you are drinking the direct product of a massive triple fusion event. Kinda wild, right?
As the seed fully matures, the surrounding ovary ripens into a fruit. Now, biologically speaking, most of the fruits we eat, things like mangoes or coconuts, they develop entirely from the ovary. These are known as true fruits. But as we know, nature absolutely loves a workaround. In some plants, the thalamus at the base of the flower actually swells up to become the fleshy part that we eat. Because they do not develop exclusively from the ovary, they are botanical imposters. Science calls them false fruits. That means your everyday apple, the strawberry, even a cashew, all technically false fruits. Who knew? To wrap up this explainer, I want to leave you with a sequence of questions from the chapter that honestly just blew my mind when I really thought about the sheer, staggering scale of plant reproduction. Picture a massive tree in full spring bloom. How many microscopic eggs are sitting inside a single embryo sac? How many of those sacs are tucked into just one single ovule? How many ovules are packed into just one ovary? And then, how many thousands of flowers are blossoming on that single tree all at once? When you start to multiply that out in your head, the reproductive capacity of a single plant is almost beyond comprehension. It's this endless, silent explosion of microscopic engineering, and it's happening right in front of us every single day. Thank you so much for joining me on this deep dive. I really hope you look at your garden a little differently today. Keep questioning, keep learning, and I'll catch you in the next explainer.